In this lecture, we will talk about the glandular tissues and also about classification of glands. The term glands actually is used to describe an aggregation of cells which are designed to produce different secretions. In general, glands are developing directly from epithelia. So what happens is that the epithelial cells invaginate into the underlying connective tissues, divide and form glands. Glands in the human body are divided into two types: the endocrine glands, which secrete directly into blood and therefore they do not contain ducts. We call them ductless glands that secrete the substances directly into blood. And then exocrine glands, which secretes the substances onto the epithelial surface from which they were derived. In this lecture, we will be talking mostly about exocrine glands and their further classification. Now, initially, we can classify exocrine glands according to the mechanism or mode of secretion. And from that, we have three separate types. The first type we call merocrine, where merely the secretion is extruded from the cell. And we see this type of secretion in the salivary glands. So, from the apical surface of the cell, only the substance, that is saliva, is secreted from these cells. Then we have another type which we call apocrine, where parts of the cell, a part of the cell or a vesicle containing the secretion is shed. This kind of secretion is seen in the mammary gland. When milk is secreted, milk is enclosed in a vesicle formed by cell membrane and secreted as liquid form. Then we have holocrine secretion, where the whole of the cell is shed. The cell containing its secretions is shed from the gland and it is the substance of secretion. This kind of secretion is seen in sebaceous glands. Then we can also talk about the types of secretion. There are two different types: serous secretion and mucous secretions. Serous secretions are watery secretions, most of the time things like enzymes. Then mucous secretion is basically thick secretions which are formed by glycoproteins, namely mucin, and it is viscid. It is quite thick. Now just look at this. This is where you have this H&E section shows you the sublingual gland, and the sublingual gland is predominantly mostly a mucous gland. Now the acini that form this gland are lined by mucus secreting cells. The mucus within the cytoplasm of these acinar cells are there in a vesicle, and inside this vesicle you get the mucus, which is called the mucin protein. Now mucin protein does not stain well with H&E. Therefore we see it as a very lightly staining area. Because the mucin is, a lot of mucin is there, the nucleus is flattened and pushed to the periphery. Okay. Then we go on to the serous glands, where the cells are serous cells and the acini are lined by these cells. The nucleus is quite prominent and ovoid, and the cytoplasm contains a lot of secretory vesicles which are again darkly stained. Now here you can see that all these cells are very darkly stained. Only the duct system is lightly stained. So this dark staining is because of the proteinaceous material which are in the secretory vesicles which stain darkly. So this is a slide H&E section of the parotid gland, which is another type of salivary gland. Then we talk about mixed glands, where you see both mucus secreting cells and serous secreting cells. So in the acini you get mucus cells and serous cells. Now because the mucus cells are much larger and the mucus granule or the vesicle is very large, the serous part of it gets pushed to the periphery and we call it a serous demilune. Serous demilune, which is an identifying feature of a mixed gland. So the submandibular gland is a mixed salivary gland, and this here you can see this section stained by H&E. This part is the mucus part, this is the serous demilune, right? Demilune means half moon shaped. Okay, so serous demilunes are seen in mixed glands. Now we will go on to more details of exocrine gland classification. Initially you can divide it into two, single cells and multicellular. So single cells, example would be a goblet cell. Now a goblet cell is a special cell which is designed to secrete mucus. On the luminal apical surface of these cells you have a large vesicle of mucus, and we call it a goblet cell, and you will be able to see them as single cell glands in respiratory epithelium and also lining the gut. Then coming on to multicellular exocrine glands, which have the most complex classification. We classify it according to the duct being either branched or unbranched. Then the secretory portion either being single or branched, and also by the shape of the secretory part, either tubular or acinar. Now here you can see a tubular gland without any branching of the duct. Here you can see a tubular gland with branching of the secretory portion. Here you see the acinar shape of the secretory part and a tubular shape of the secretory part. Now let us first talk about simple glands. The duct of these glands are unbranched and directly opens onto the luminal surface of the epithelium. Simple glands are further subdivided according to the shape of the secretory portion and also by the branching or non-branching of the secretory portion. Therefore, in humans, we see three different types of glands. Simple tubular glands, two types, simple branched tubular and simple coiled tubular. Then simple acinar we might see, and also simple branched acinar. Now talking about simple tubular glands, the secretory portion is tubular in shape and is not branched. So we have an unbranched duct ending in a tubular secretory portion. This unbranched duct opens directly onto the luminal surface or the epithelial surface. And we see simple tubular glands in the mucus secreting glands of the colon, the mucus secreting glands of the fundus and the body of the stomach, and also in the endometrium of the uterus, where we call them uterine glands. Coming on to simple branched tubular glands, the duct of the gland is unbranched and opens onto the luminal surface, but the tubular secretory portion is branched. So you have an unbranched duct ending in a branched tubular portion, and the glands in the pyloric region of the stomach are of this type. Glands in the pyloric region of the stomach are simple branched tubular. Then going on to simple coiled tubular, where again you have an unbranched duct and the tubular secretory portion is coiled. This you see in sweat glands. Now, because of the coiling, when you take a cross section like what you see here in the H&E section, you see longitudinally cut tubes, and also the tube cut in cross section, circles and sort of ovoid areas. That is because this is a coiled tubular part of the secretory part. Then simple acinar glands are rather uncommon. So you will have a short unbranched duct opening onto the luminal surface, and the duct will end in an acinar secretory portion. Sometimes it's difficult to figure out whether it's a simple acinar or a branched one. Okay, so mucus secreting glands of the penile urethra consists of these simple acinar type of glands. Then simple branched acinar, that is where the unbranched duct ends in branched acinar secretory portion, where you can see this is a sebaceous gland. The duct is coming down like this, and the secretory acinar portion is branched. Okay, so sebaceous glands of the skin is an example for simple branched acinar glands. Then going on to compound glands, where the duct is branched. Then the secretory shape of the secretory portion, tubular and acinar, and you can have a mixture of these two, which we call tubulo-acinar. And also there can be branching of the secretory portion, where you would call it a branched tubular. So the name would be compound branch tubular. Talking about compound branch tubular glands, the duct is branched and the secretory portion, which is tubular, is also branched. So here we can give the example, the Brunner's glands of the duodenum. In this H&E section of the duodenum, this whole of this part is the Brunner's glands. They are lying underneath the muscularis mucosae, and with tiny ducts they are secreting, branched ducts, they are secreting the mucus onto the surface of the duodenum lining. So this is compound branch tubular glands. Then compound acinar glands, again the duct is branched several times, and each of these duct opens into a secretory acinar portion. This kind of gland is seen in the pancreas. You can see the secretory acini of the pancreas and cross sections of the duct in this H&E section. So the pancreas is an example for compound acinar glands. Then compound tubulo-acinar gland, again the duct is branched and the secretory portion consists of both tubules and acini. And we see this in the prostate gland as well as in mixed salivary glands. Now here is a H&E section of the prostate, where you can see the neuromuscular stroma and also the glandular elements, which are compound tubulo-acinar in type. Another compound tubulo-acinar gland is a mixed salivary gland. In general, the mucous cells form the tubular parts of the gland, while serous cells form the acinar part. So serous demilunes are also seen in the secretory part because there are mixed tubular and acinar portions in that particular acini. Okay, so in general, the tubular shape, actually tubular shaped glands will secrete mucus, while acinar shaped glands will secrete serous secretions. Now just a little bit about endocrine glands, which you will learn later. Endocrine glands consist of clumps or cords of secretory cells which are surrounded by a basement membrane. They are special in that they have a rich network of capillaries, and they secretions are released directly into blood, and there are no ducts. Now here, this is a H&E section of the thyroid gland where you can see lobules of thyroid cells which are formed by follicles. Inside you have the colloid, and surrounding that a simple cuboidal epithelium of follicular cells. And in between you have connective tissues, and also you can see cross sections of blood vessels.